hear you.
Thank you very much. Thank you for the kind introduction, and thank you for the opportunity to present my work today. We are very interested in working on the protein tau and how it's involved with more than 20 neurodegenerative disorders, and that's how I became acquainted with Ioannis. If maybe less than a year ago, we had a very good conversation and started a collaboration. And so I'd like to share some of the work that we've done in our lab and that precipitated our collaboration. And so the title of my talk today will be presented shortly as I share the screen.
It's very good to see the space, the Earth
I am
from the
in
space,
space.
anyhow. Here it is. Thank you.
Okay, so I'm originally from Colombia, and that's why I like to put my titles with a little bit of Spanish, also because we're talking about translation. And in this case, there's a little bit of maladaptive RNA translation in tauopathies is what I'm getting at. And the idea is to introduce a little bit of drama of tauopathies and other demons. This is inspired by the book from my compatriot, Gabriel Garcia Marquez, who wrote Of Love and Other Demons, and he ended up winning the Nobel Prize in Literature for his book, 100 Years of Solitude. And so here, taking this as an introduction, a little bit about myself and what I do, we focus on studying tauopathies. Our big ambition is to effectively treat patients with tauopathies. The goal is to expand our understanding of tau biology in order to design new and effective therapeutic strategies. And the tactic to do this is to use broad experimental strategies from cell-free assays all the way to human tissues to identify how tau drives maladaptive translation and how can we take these results to the clinic. So today I'm going to give an introduction of the work that we do, and along the talk I'll discuss our findings on pathologic tau, how it reduces translation, both in vitro and then in vivo. And that gave us interesting and contrasting findings. We also found that tau promotes a selective form of translation. I'll be talking to this point about how tau interacts with ribosomes, but then focusing on tau and RNA. And finally, what can we do about this, both in tauopathies and in a particular component of my research program, which is traumatic brain injury. So to introduce, we all are familiar with the concept of dementia. It is a decline in mental activity, and it's defined by changes in personality, social behavior, language, difficulties in thinking, making decisions, poor coordination, and balance. Tauopathies are the most common form of dementias. Many of them are Alzheimer's. And here are two of the best described cases of individuals with dementia. They have very extensive clinical histories. Here we have Don Quixote de la Mancha, and here King Lear, where two of the greatest authors in history have written extensively about some of the symptoms of dementia. In talking about tauopathies being the most common form of dementia, we have more than 20 neurodegenerative disorders, and all of these share the same pathological hallmark, which is the abnormal aggregation of the protein tau inside of brain cells. We see that patterns can be different in Alzheimer's disease, corticobasal degeneration, which is one form of frontotemporal dementia, progressive supranuclear palsy, another form of frontotemporal dementia, and chronic traumatic encephalopathy, which is the end result of repetitive mild head injuries. One way to describe what Alzheimer's looks like is beyond the clinic. And I like to present this particular slide of an artist whose name is William Utermohlen. Here is a very gifted artist. In 1967, he drew a self-portrait, and he was diagnosed with Alzheimer's disease in 1995. And then throughout every year, he vowed to make different self-portraits. And you can see how there's a tremendous amount of changes that could be the manifestation of motor changes, of cognitive changes. The darker colors could be a manifestation of depression. And all of these manifestations are true and common in many patients suffering from Alzheimer's disease. And a more clinical component, Alois Alzheimer was both a psychiatrist and a neuropathologist. And in 1906, he described pathologically the brain of a patient, August Dieter, with these two pathological hallmarks, which are the extracellular aggregates of amyloid plaques, which are composed of a small peptide called amyloid beta, and the intracellular, and in Alzheimer's in particular, intraneuronal aggregation of the microtubule-associated protein tau. This is what we focus on. Here, this protein tau builds inside of neurons, and colloquially speaking, it seems to impair that neuronal function, almost like choking and preventing transport along that neuron, affecting neuritic function, synapses, etc. Tau was discovered back in the mid to late 70s by Don Cleveland and Mark Kirschner, and is identified as a major microtubule binding protein in the brain. Tau is a fascinating protein which has 441 amino acids, and throughout you can see not only that it has splicing events, making it post-transcriptionally modified. Here, these two exons 2 and 3 can be spliced out, and exon 10 can be spliced out, leading to 6 different isoforms. Tau is also post-translationally modified by hyperphosphorylation. It's phosphorylated in many sites, glycosylation, nitration, ubiquitination, acetylation, methylation, and also cleavage at numerous sites, leading to fragments, some of these having toxic properties. But tau to this point, and what we know about disease, is typically put in a light of a negative or a malicious protein. It has a deleterious effect. But tau has a very important function in the cell. Here we have our neurons that have this very svelte and conformation. They have beautiful extensions, in this case an axon, but that presents a unique challenge. That is that if cargo needs to be transported from one side of the cell, for example, a mitochondrion, to the other side of the cell, then it requires a unique transport system. This is based on microtubules. For what we know from microtubules, alpha and beta tubulin form polymers, and tau binds to the polymers, stabilizing those microtubules, and now building what would be a bridge. And then, thanks to motor proteins, the mitochondrion can be now transported from one side of the cell to the other. And this is all works very nicely and exquisitely regulated inside the cell. The challenge now is that the cytoplasm of a neuron is extremely crowded. And as a result, this dynamic microtubule needs to disassemble. For that to happen, tau is acted upon by kinases. It's phosphorylated, diminishing its affinity for the microtubule. Now the microtubule disassembles. But here's the problem. Tau is biochemically defined as an intrinsically disordered protein. That is to say, it is not stably formed in a particular fold. And so it is highly hydrophilic in the cytoplasm, and it can adopt some conformations that can now begin to adopt a toxic fold. And that leads to this soluble oligomeric tau conformers. While we don't know how these processes occur or how the toxicity takes place, what we do know in the brain of a patient with a tauopathy, in particular with Alzheimer's disease, is that tau is hyperphosphorylated and it aggregates surrounding the nucleus around the soma. Now, everything to this point, and tau is best known as a microtubule stabilizing protein. However, work from many labs, including our own, have identified a tau interactome. Many of the proteins that interact with tau are far beyond microtubules. In fact, from our work, we took human brains, either Alzheimer's or control. We isolated the endoplasmic reticulum and did co-immunoprecipitations, where we pulled down either tau or actin. We ran these on a gel and we did mass spectrometry because we wanted to know which proteins associate with tau. And in 2 separate papers, we published our findings that a large proportion of the proteins that associate with tau participate in translation. In fact, many of the proteins that associate with tau in Alzheimer's disease are different from those in control brains. And there are far more of RNA binding proteins that associate with tau in Alzheimer's than in the controls. Now, the association of tau with ribosomes had been described in the late 80s by Skip Binder's lab. But for more than 30 years, the consequences of the tau ribosome interaction were unknown until 2016, when we published our work showing that pathological tau reduces ribosomal function in vitro. For this study, we used 3 different in vitro models. We used a cell-free assay to make sure that whatever interaction occurred between tau and ribosomes was directly the response of that interaction and not any kind of compensation by the cell. We used an immortalized cell line, and I won't show these results for the sake of time. And also we did primary neurons, particularly from tau transgenic mice. Our hypothesis was that under normal conditions, tau associates with ribosomes, leading to normal translation. Under pathological conditions, that pathological tau species, hyperphosphorylated, bound to ribosomes and prevented translation. Our first experiment was to corroborate the findings that tau associated with ribosomes. To this end, we just did separation of proteins, and I will show you the data from a whole cell lysate, a fraction that does not have ribosomes, and a fraction that is enriched for ribosomes. In this Western blot, I'm showing you data from our control brains, our Alzheimer's brains, and our fractions with the whole brain lysate, a fraction without ribosomes, and a fraction with only ribosomes. And you can see that the most dramatic difference is that tau associated much more strongly with a fraction that only had ribosomes in Alzheimer's disease compared to the control. In fact, a lot of this high molecular weight protein on this Western blot corresponds to a hyperphosphorylated pathological tau species called paired helical filaments. And these are identified with this antibody PHF1. Here we're using 2 antibodies for ribosomal proteins to confirm that our separation was done properly. You can see that the ribosomal proteins P0 and L28 were only enriched in the fraction that had ribosomes, and that HSP70, which is a protein that is primarily cytoplasmic, was not present in the ribosomal fraction. So we did this experiment with the proper controls. In addition, we did immunofluorescence staining of human Alzheimer's brains, and we find that tau oligomers, which are the toxic tau conformers, highly toxic, stained in red, overlap with this ribosomal protein S6, suggesting that toxic tau species interact with ribosomal proteins. So again, we've confirmed what had been shown before. So what are the consequences of this? Our first experiment here was using a cell-free assay where we had a 96-well plate or tubes where we added all of the machinery necessary for translation to occur. That is to say, ribosomes, tRNA, initiation factors, elongation factors, ATP, etc. We added a plasmid for GFP, so that way we can see translation and folding of a new protein. And we could add tau to some of the wells or other controls. Here are the data. Over the course of 6 hours, we see that GFP levels, representative of new proteins, were increased in the wells that had BSA. But when we added tau oligomers, the amount of GFP, the fluorescence, was significantly decreased by 40%, suggesting that tau oligomers interact with ribosomes and reduce its ability to produce new proteins. We then sought to establish whether this was the direct result of tau or any oligomeric protein. And so we did an experiment now using synuclein oligomers, alpha-synuclein, or insulin oligomers. And we saw no change in the amount of GFP produced compared to BSA. So these data suggest that tau is responsible for reducing translation in this in vitro model. But why would the cell care about that? So here we did primary neurons in this experiment. We obtained mice from Dr. Jada Lewis. These are the RTG 4510 tau transgenic mice. They overexpress a human mutant form of tau that is associated with frontotemporal dementia. There are issues with the mouse in that it was at the time, the transgenes interfere with expression of other genes. But importantly, we generated primary neurons from the brains of these mice with the ultimate objective of being able to establish, is translation affected in primary neurons from tau transgenic mice? And for this, we used a technique called sunset. Sunset, or surface sensing of translation, is based on the principle that puromycin, an antibiotic, is structurally similar to tRNA. So when we add puromycin to a cell culture, it comes in and in some way fools the ribosome and adds a puromycin tag to any new protein. So now new peptides have a puromycin tag, and we can now detect these proteins with an anti-puromycin antibody. And here are the results of our data. Our non-transgenic cells have puromycin levels showing the normal amount of translation, but our tau transgenic cells had a significant reduction in puromycin, suggesting that translation is reduced. In fact, one of the proteins that was reduced is PSD95, which is an important protein for synaptic function. Therefore, these cells that overexpress tau already have a structural impairment in synaptic function. This is to show that the cells overexpress tau. Here's a quantification. Puromycin levels dropped by almost 50%, almost 50% reduction, and PSD95 levels decreased by almost 90%. But it's hard to say whether this is the result of translation or transcription. So we looked at RNA for PSD95, and we found that in the transgenics, there was much more RNA. So what we have is a lot of RNA and very little protein, suggesting that translation is impaired. So these data corroborate that our hypothesis, which is a pathological tau species associated with ribosomes and reduce translation. But does this happen in vivo? So we went back to the mice, and I have to note that these mice were very astutely generated because they have a regulation system. When we add tetracycline to the diet, it turns off tau expression. So when we add this drug to the water and the diet, the food, the mice stop making tau. So we looked at the mice at 5 months and at 7 months. And we did a puromycin assay where we injected puromycin in the body, it went to the brain, and there it tagged all new proteins. And we did immunohistochemistry and quantified that and found that at 5 months, there's a reduction in puromycin in translation. But that reduction was not significantly different. At 7 months, we saw a stark and significant difference in the amount of protein synthesis. But this is problematic because at 5 months, these mice already have cognitive deficits. So we wanted to see, well, is the reduction in translation responsible for cognitive impairment, or is it that there's a change in translation that is responsible for that cognitive impairment? So we looked earlier, and this is a cool experiment where we treated the mice with doxycycline to create a rescue experiment. This is a mouse timeline. By 2 to 3 weeks, they start expressing tau, human mutant tau. By 2 months, you start seeing early pathology. By this time point, between 2.5 and 5, there are many reports showing cognitive impairment and deficits in neuronal plasticity. The neurons are not firing correctly. And finally, by 5 months, there's significant neuronal loss. There's tremendous brain atrophy. We took one group of mice and treated them with doxycycline at this time point, before there was significant neuronal loss. And we used it as a control. And at this point, we injected with puromycin. We took half the brain to measure RNA, and we took the other half to measure protein. But we were very interested in proteins that are newly synthesized, which again tells us a lot about translation. So again, we'll have a lot of information on RNA, we'll have a lot of information on protein. These are the control results. These are non-transgenic mice, either treated with a vehicle or doxycycline or tetracycline. To turn it off, there's no change in tau. Here are the tau transgenic mice showing robust expression, and then a 50% reduction in tau accumulation in the tau transgenic mice treated with doxycycline. So that suppression of tau worked well. These mice actually get better and cognitively improve, showing that this is a good control of a rescue. In terms of the RNA, we did microarrays, and we found 3 different patterns of expression. In non-transgenic mice, we found that these RNAs were not significantly changed. But the tau transgenic mice that overexpressed tau had a substantial change in these transcripts. And when we suppressed tau expression, their expression went back to normal. 2 other patterns were some that doxycycline didn't do anything to it, some that had an effect of doxycycline. All of these are the appropriate controls, but we are very interested in the transcription of proteins involved in translation because they associate with tau and they're most likely responsible for changes in translation. And so we looked at a number of these ribosomal proteins or proteins involved in initiation, translation, or elongation, and we saw no significant difference in expression in transcription. Therefore, any changes that we see at the protein level is not the result of transcription of the RNA. It is the result of protein synthesis. So here are the results of the proteomics. First, we injected puromycin, once again, with intraperitoneally. We pulled down with immunoprecipitations and did tandem mass spectrometry to identify all the peptides associated with puromycin. We found no significant differences between the samples that were non-transgenic, whether treated with a vehicle or doxycycline. We found that many proteins involved in translation were reduced in the transgenic mice. In this gene ontology class, all the proteins that were involved in translation ribosomal were, and this is a relative value, significantly reduced. But then in the mice, where we suppressed tau expression, that we would expect an improvement in cognition and a reduction in tau pathology, we saw a massive upregulation or rescue in this group. So this tells us that tau expression is responsible to a certain extent, we don't know how, to regulate translation of ribosomal proteins. And so that led us to the hypothesis that tau promotes selective translation. And we did this with the further corollary that tau may do this by interacting with ribosomal proteins. So I'm going to go back to these data showing that tau oligomers interact with this ribosomal protein S6. And I go back to it because that's the one that we focused on. And it became a very interesting protein. Ribosomal protein S6 has properties that allow it to regulate which proteins are synthesized at any particular time. The active form of phospho-S6 promotes translation of proteins involved in cell growth. It will promote translation of 5' top mRNAs. These RNAs code for all ribosomal proteins, all the initiation factors, and some elongation factors. In other words, when a cell is happy and growing, it needs more ribosomes. So at this point, S6 is phosphorylated and more ribosomes can be manufactured. We did our first level of experiments in vitro. We used this inducible cell line of human embryonic kidney cells that overexpress tau. These cells, when we add tetracycline to the media, it turns the system on and it turns tau expression on. And here you can see when we add tetracycline, after 1 day, 24 hours, there's already tau expression. And tau levels go up over 4 days of exposure. And so here are the results of our first experiment. We induced tetracycline for tau expression for 1 day, 24 hours, and we see that pathological tau and total tau levels were increased, as we would expect. Over the course of 4 days, we had much more expression of pathological and total tau, or accumulation of pathological tau. And then we wash off the media and we don't add tetracycline. So now the system is turned off for 1 day, and tau levels are reduced, and eventually they go back to normal, to no expression. So this is a wash or rescue experiment, similar to the mice. Here we're showing that the total levels of active levels of the ribosomal protein S6 are not changed after 1 day of tau expression, but after 4 days, the phosphoform of S6, the active form, was virtually undetectable, suggesting that there might be a change in translation dependent on S6 and driven by pathological tau. Because when we get rid of tau, phospho S6 comes back to normal. There's this inverse correlation between phospho-tau levels and phospho S6. To establish whether this relationship was important in human brains, we did immunoprecipitation assays where we isolated with an antibody S6 or tau in human control brains or Alzheimer's brains. These are late-stage Alzheimer's brains. And we found that both tau and DIS6 co-immunoprecipitated, so they're both associating together. We also found that this association was more robust in the Alzheimer's brains. Once again, here we have the quantification of specific 5' top mRNAs. Here we have ribosomal protein S6, initiation factors, and there are the protein levels this is RNA. RNA levels continue to increase, as we saw before for PSD95. There's a lot of RNA, but when we look at the protein, there's a significant downregulation or reduction in S6 and each one of the proteins that are coded by these genes. In other words, once again, now in vivo, in a human brain, there's a lot of RNA for these proteins and very little protein. So it's hard to think about translation without considering RNA. Tau has been recently identified, and this is work that has been pioneered by Dr. Ben Wolozin, and I've had the opportunity to collaborate with Ben on some of this work, showing that tau interacts with RNA. And these tau RNA complexes have been identified many years ago, showing that they may form these liquid-liquid phase structures inside of the cell, these aggregates that are characteristic of cells that have tau pathology. We asked, okay, if tau associates with RNA, then which RNA is associated with tau? For that experiment, we took human Alzheimer's brains and controls, and we did an experiment called enhanced cross-linking and immunoprecipitation. For this, we took an antibody that detects tau and an antibody that detects hyperphosphorylated tau, and we did RNA-seq from human brains. This is the antibody that has really high affinity. It was developed by my colleague here at UF, Dr. Benoit Giasson. And these tangles or aggregates are identified by this antibody 7F2. We used different types of human brains controls, which are this early primary age-related tauopathy. We used low-stage Alzheimer's disease and late-stage or high AD. And these are the results. When we pulled down tau from these brains, we did RNA-seq, we found that there was a shift. The vast majority of proteins of RNAs that are coding, that is to say, messenger RNAs that associate with tau, in blue, increased with disease progression. Therefore, there's more association of tau with coding RNAs as disease progresses. And all this came at the expense of a reduction in tau association with non-coding RNAs. And this is even more pronounced when we isolated pathological tau. This is in the low stage and in the high stage of Alzheimer's disease. In blue, we see a massive upregulation or increase in the types of messenger RNAs that associate with tau. So again, it confirms that there's a lot more coding RNA that associates with tau with disease. And again, that comes at the expense of non-coding RNA interactions. To establish which RNAs these are, we started looking at consensus sequences. And we found some very interesting things, that controls had some consensus sequences that were enriched in the Alzheimer's disease brains. And when we started mapping these out, we had an N of 4 per group. We started finding this pattern of UGA repeats that were far more frequent in the samples we found for Alzheimer's disease. So this UGA sequence seemed to have higher affinity for tau in Alzheimer's disease. When we mapped this out with different databases generated by Drs. Todd Goldie and Karen McFarland, now at Emory University, we found that some of these sequences associated with tau were non-coding RNAs, and that was surprising. Now we have an enrichment of RNAs that associate with tau in disease. And these are particularly non-coding RNAs that regulate translation. In fact, they regulate translation of, for example, RNA polymerases, which are critical for protein synthesis. So can we rescue these effects? We already know that there's a functional deficit. Can we restore translation with the expectation of rescue and function? For that strategy, we worked on a protein called PERK. And we're interested in this protein called PERK because its job is to reduce translation. The way that PERK functions is that it is it protects the endoplasmic reticulum. So here we have the nucleus of a cell, the endoplasmic reticulum. These are ribosomes showing the rough endoplasmic reticulum. And here I'm showing the proteasome. The endoplasmic reticulum, or the ER, provides the optimal environment for proteins to fold properly. Here we have an example of a membrane protein that is folded and goes to the membrane to do its job. But not every protein will have the same fate. Here we have this black ribbon protein that did not fold properly. It will be ubiquitinated and targeted for degradation in a process called endoplasmic reticulum associated degradation, or ERAD. In this process, certain carrier proteins such as VCP or P97 pick up that ubiquitinated protein and take it to the proteasome where it is degraded. What we found in 2013 is that tau blocks ERAD, promoting an accumulation of ubiquitinated proteins around the ER. Imagine there's a bulk stop of proteins exiting the endoplasmic reticulum, and that induces ER stress. ER stress is resolved by autophosphorylation of this protein PERK, and PERK reduces translation of new proteins so that if you have less protein input, then the endoplasmic reticulum can resolve its traffic jam. This protein PERK was very interesting because it had already been genetically associated with tauopathies. The largest genome-wide association study for this disorder, progressive supranuclear palsy, or PSP, identified SNPs on PERK, on the gene coding for PERK. We have found it molecularly, that link, and now there is also a pathological link from the late 2000s, 2009, showing that there's more phospho-PERK in Alzheimer's brains and later on in frontotemporal dementias, showing that this protein PERK is heavily involved. And since PERK regulates translation, then once again, translation is affected in these disorders. Normally, PERK, which is coded by the EIF2 alpha kinase 3 gene, targets this initiation factor subunit, EIF2 alpha. And when this is phosphorylated, it doesn't work very well, reducing translation. What happens is that's good to restore translation short term. But when this happens long term, you have much more phospho-PERK, you get a pathway that says the cell cannot cope with the stress and it induces apoptosis. So we hypothesized that if pathological tau species, since pathological tau species induce ER stress, which results in a long-term upregulation of phospho-PERK, and that in and of itself reduces neural function, then we could rescue that by inhibiting PERK. And so we found a chemical inhibitor called GSK 2606414. To summarize to this point, PERK is an unfolded protein response kinase that helps restore ER function by suppressing translation. We found already that tau activates PERK. This drug is developed to inhibit PERK, and it's important to note it's an ATPase inhibitor. And as a result, high concentration of this drug can interfere with many other kinases. So here we have some of the preliminary data showing that this drug could engage and have an effect. In RTG4510 mice, tau transgenic mice that have profound deficits in translation at 8 months, treated with the drug, here's untreated with vehicle, puromycin in green, and when we treat it for 2 days, there's a substantial rescue in the amount of protein. So much more protein is produced when you treat with this inhibitor. This tells us that the inhibitor can rescue translation in these mice, and now we could have a potential therapeutic strategy. To test this, we used the 4510 mice once again, and here, instead of at 6 months, we know that PERK levels activate after 6 months. So what we sought to do was to inhibit before that as a treatment, a therapeutic intervention that would be preventative. So we treated the mice with the inhibitor, and we did MRI, magnetic resonance imaging. These are T2 to tell us anatomical measurements, and we found that after 1 month of treatment with the drug, there is already in the transgenic mice, here we have with vehicle, we had brain atrophy in different areas. This atrophy in the volume here, we can measure it, was prevented with the drug 414. In other words, here are non-transgenic mice, that's a normal volume. With the transgenic, there's a decrease in the volume that is prevented by treatment with the drug. So we thought this 1 month time point could be interesting. So then we measured function. And one way to measure function using MRI was using this approach called manganese-enhanced MRI. And manganese is very helpful because it is similar in size and identical in charge to calcium. So it enters voltage-gated calcium channels. When calcium comes into the cytoplasm, it allows for neurotransmitter release. It substitutes calcium, and more importantly, it is paramagnetic. So now we can visualize where manganese is and how it flows in the brain with the MRI. This is what the manganese-enhanced MRI looks like. At baseline with no manganese, you get the signature. When you inject manganese chloride and wait 6 hours, you get this signal in that particular mouse. And here are the results of our experiments. Here we have our non-transgenic brains treated with a vehicle or the inhibitor, and the brains look very similar. There's no difference. Here's the transgenic treated with a vehicle. You can see that the hippocampus is much, much thinner. There's brain atrophy. In fact, this blue spot here defines brain atrophy. This is CSF. This is fluid. Because the brain in this mouse that's transgenic, overexpressing tau, has so much damage. But this damage is prevented when we treated the mouse with the inhibitor. You can see the hippocampus is full. There's limited to no brain atrophy. When we did quantification of the flux in the reciprocal R1 of the T1, we saw some decrease, but not significant decrease in the dentate gyrus of the signal or in the CA1. There was a massive deficit in the transgenics in these both brain areas that was rescued or prevented by the drug. We did other measures of function. For example, this is the open field maze. Here you place the mouse in the middle of a box. If the mouse is on the edges of the box, then it represents a phenotype of anxiety. And the red lines trace where that mouse is running. This seems to be defined as an anxious mouse. Now, when we treated the mouse with the inhibitor, it didn't seem to care where the edge was. It was just very relaxed, no problem, when it was treated with the inhibitor. We also did behavior. This is using the novel arm test, where mice have this natural tendency to explore the new arm of a maze, and this is a Y maze. And here you can see that the vehicle-treated transgenic mice had a massive deficit. This is, in some ways, limitation of that exploratory behavior that is rescued with the inhibitor. When we went to study mechanism, we were not able to identify whether this was because of tau or not. In fact, we found that it is not because of tau. Tau levels didn't change. And this is really important because this tells us that here we have an intervention that can help cognition, whether the patient or the mouse in this case has tau pathology or not. In other words, it's not too late to treat this patient or this mouse. The problem is that at this concentration, this inhibitor is not extremely specific. It can also inhibit 22 other kinases. So doing mass spectrometry here with tandem mass tagging, we tried to find which ones could be possible avenues, and looking at different pathways, we found that sure enough, translation was rescued. We doubled the amount of translation that was deficient in the mice, and we narrowed down to 4 proteins that we are currently investigating. And this is by cross-referencing multiple databases and finding that pathways in which these proteins are involved could be responsible for rescuing. These studies are now underway in the lab. So let's shift gears. I want to talk a little bit about TBI and translation. My lab is very interested in repetitive mild TBIs. There is a clear association between traumatic brain injuries and neurodegenerative disorders. 1.7 million people in the United States sustain a head injury every year. And this number is only those who are reported. People who sustain concussions or mild head injuries hardly have any immediate neurological symptoms, so they never go to the hospital and don't report it. That seems to be a latent problem, because if these individuals have a head injury and later develop neurodegeneration, we won't be able to detect whether this is a comorbidity or not. One of my favorite sports is football, soccer, and I've been doing some research onto if there's an increase. There is, in fact, an increase in soccer players dying of a neurodegenerative disorder. There's an increased risk, and many of them develop dementia. Defenders seem to do worse in dementia because they have more headers. That's the hypothesis. And the magnitude is expected to be much greater. The problem understanding TBI, and one of the bigger limitations, is that it's unpredictable. If a soccer player hits the ball with a header, a football player, you'll have vectors of force that transfer from the front to the back. That's the coup effect. But then there's a contra coup effect, where vectors of force are going to spread throughout the brain in different directions in ways that are hard to predict. In fact, rotational damage is very difficult to predict. TBIs here, borrowing from Emory University, is considered a disease of process. An event early on after the injury can lead to secondary injuries throughout the life of that individual patient that may not be immediately recognizable. So it's very important to establish biomarkers. Here we have imaging biomarkers with TAC or CAT scans, computer-assisted tomography, and they're very inconsistent. It's hard to tell whether a person will recover well or the prognosis of that patient is going to be worse. And the same with protein markers. Here you can see that in the order of minutes, hours, days, weeks, and months, there are massive oscillations in the accumulation of proteins, different proteins. For example, tau. Tau spikes early in the matter of days and then drops and then spikes again. This is very difficult to use as a biomarker for prognosis. So in my lab, we're interested in identifying biomarkers, which can tell us about mechanisms and ultimately identify therapeutic targets. The model we use is called the closed head injury model of engineered rotational acceleration, or chimera. This model, here we place the mouse. There's a piston at the bottom. It hits the mouse at the top of the head, and we see remarkable consistency. Here we have quantification. As an example, the optic tract of accumulation of here IBA1 and here GFAP. This is a marker of microglial activation. This is a marker of astrocytic reactivity. In our first experiment, we wanted to see what are the most immediate changes we can detect. And we found that after 2 injuries, because it's repetitive, we take a mouse 2.5 months and injure them one day, 24 hours later, we injure again. And then 15 minutes, 30 minutes later, we collect the brains and do immunohistochemistry. And we found, using pure myosin, that there are many areas of the brain where there is less translation. Here we have this area of the brain as an example, where there's a reduction. And we hypothesize, is this because of PERK? Here we have data from another model showing that after 30 minutes, there's substantial upregulation of PERK, 30 minutes and 3 hours after the injury. And this is the hippocampus. These are neurons. This is the neuronal layer of CA3 and CA1. And here's the dentate gyrus. You see there's a massive upregulation of phospho-PERK. And so we stained for phospho-PERK in our model, in our mice, and we see that areas that coincided with low translation had high levels of phospho-PERK. So maybe PERK could be targeted in our model. So instead of going to the drug to use it as a therapy, we wanted to be sure that it would be specific to PERK. So we used this transgenic model. These are PERK knockdown mice, and the Crete were controlled. And here we used the influenza variable of age. Because patients in later stage of life have more falls, they're at higher risk of falls and repetitive falls, we wanted to focus on older mice. And here we did 2 injuries at 12 months of age, and at 7 days, we collected the tissue. In between, we did now resting state functional MRIs. The resting state functional MRI tells us now about function based on oxygen levels. Very briefly, it can detect oscillation of water and the change in water as the basis of and the exchange of O2 for CO2. So the paramagnetic properties of oxygen are different from those of CO2. And so that tells us that when there's consumption of oxygen, we can read that change throughout the brain. And then we couple these data with graph theory so that we can place nodes after the measurement in different brain regions. And now we can make mathematical calculations to establish relationships between those nodes. The way that I explain this and wrap it around my head is by understanding that, for example, each one of these nodes in the United States represent an airport. And what we're trying to figure out is what happens when an airport like Atlanta gets slowed down. There's an impact, there's damage, there's traffic. Then there's rerouting of the entire brain, of the entire airport traffic in the United States. And so now we can measure things such as how many flights are rerouted to other airports, how many are slowed down, as numbers such as synaptic strength or the length of these connections. One of the measures that we can do, and I'm going to go through this very briefly just to show that we can measure things like clustering coefficient, local efficiency, and node strength. And many of these are reduced after TBI. You can see that in different brain regions, a lot of it in the thalamus. And we see that in other areas. There's local efficiency as a second measure also decreased. Interestingly, node strength increases, and that could be because of compensatory mechanisms, rerouting of those signals to different nodes. When we did the experiment in the PERK knockdown mice, we would expect a recovery. And indeed, we found that, for example, clustering coefficient in the visual postfrontal cortex and the posterolateral, there's a very low signal in the controls, whether injured or not, that is the Crete littermate control or the PERK knockdown. And when we injured them, there was an upregulation of the clustering coefficient, these 2 cases, that was rescued in the PERK knockdown. And we saw the same phenotype for local efficiency in the postfrontal cortex. There was here a decrease in the signal that was rescued in the PERK knockdowns. We also did nanostring to measure changes in protein levels. And this is essentially doing a series of immunohistochemistry experiments where we can detect 72 proteins at once. And this is spatial. So when we look at, for example, the optic tract, we saw upregulation or an increase in the accumulation of proteins, such as phosphatidylserine 199, CTSD. Very nice to see that IBA1 and GFAP also went up because that's what we saw and validated from our immunohistochemistry experiments. But other proteins went down. So it gives us a good sense. And now we can start measuring things in the PERK knockdown mice. And we see that many of the proteins that changed are involved in inflammation. PERK knockdown mice had a reduction in these inflammatory markers, which suggests that there might be a rescue. However, not all the proteins that we would expect to be beneficial were rescued in the PERK knockdowns. Here we have myelin basic protein. We would expect a recovery in myelin, but in fact, we found that the PERK knockdown mice have less myelin. So we're not entirely sure whether PERK is a good therapeutic target at this point, but it's certainly promising in revealing new therapeutic avenues. We're doing other work, functional connectivity matrices, where we hope to be able to put this at every patient's bedside on their chart. If we can narrow down which brain regions have different measurements that are active or inactive, we would be able to then measure changes in prognosis. We can look at the entire brain. We're increasing different nodes throughout the entire brain. We're doing metabolomics. We're finding accumulation of glycogen in different brain regions. Lipidomics are different between different brain regions, whether injured or control. And I'm going to skip through the conclusions because I'm running low on time. But of course, I want to show you that I feel like I downplay the importance of sports. And sports provide many health benefits. And I'm not saying that we should stop playing contact sports. I think we need to understand and mitigate that. Here we have, for example, this is my 5-year-old son who's playing in his team, and he crosses the ball, and he's going to be great, but he passed the ball to nobody. And here we have another image of a neuroscientist who does this amazing, spectacular header, just to show that this is more of a public service thing. It's important to understand these risks of playing sports, but it doesn't mean that we should stop playing these sports. And so with that, I'll acknowledge the lab, everyone here at the University of Florida, my colleagues, the University of Kentucky, many folks who've shared brains with us, Emory. We have collaborations with the PERK inhibitor and Sapienza University, our colleagues, GlaxoSmithKline, and my former mentor who passed away, Chad Dickey. And with that, I thank you all for your attention.
Great talk. Great work.
Thank you, Josh.
Thank you very much.
Fantastic.
Do you want me to stop sharing?
Fantastic work.
Thank
Yes,
you.
you can stop sharing. Thanks. This is great work.
Thank you very
Once
much.
more, we are impressed by the work and the multi-level analysis you are using in the lab, from animal models, cell culture to animal models, and then to AD brain, which indeed is a great pipeline to understand and go back to treatment, to find targets and go back to treat.
Thank you very much.
So, is there any questions? I have plenty, but I will leave the floor. I'm sure the people from YouTube, which usually are the most of the attendants, use the YouTube to see the seminars. Do you have questions from the people that are here or from the YouTube? If not, so I could. Ioannis, do you see something on anything on the YouTube?
No, go ahead, John. Go ahead, John.
Okay.
I have some questions, but later.
So okay. What is your view on what happens in the, if we try to understand the two ways that tau affects neurons in the AD brain through the process, even through the 10 or 20 years preclinical phase before we have the first symptoms. Do you think that we have a loss of normal function of tau that affects more based on your data, or it is the presence of the accumulation of pathological forms that affect more translational transcription? Or do you have an idea of what is first or what is second? Because some of your work was developed on the late Azlheimer disease brain, but some you can see also early. So do you have a scenario of loss of function versus gain of toxic function, in which part and where translation or transcriptions are affected? I know it's a huge question, but I want to see your vision. You are many years in the field.
I'll answer very briefly. I think that tau has a normal function in regulating translation, and under stress conditions, it facilitates translation of proteins that will participate in adaptation. I think under these disease processes, there's a stress, for example, in TBI, and tau helps the system make proteins that will resolve the stress event. I think that when it's chronic, and tau is active for too long in doing that, it now promotes translation of proteins that should not be made, that downregulate the system. For example, a neuron under stress should be conserving energy. Should it be firing, making a lot of synaptic proteins, new ribosomes? No, it shouldn't. It should be conserving energy at a metabolic level to be able to repair itself. But 20 years of this is not sustainable. So that's where I think there's a gain of toxic function.
Okay. Would you consider that you have, for example, a tau knockout explanation to your experiments? If you use tau knockout, because we know, for example, Deitner work that using TBI, tau knockout, and he see that there was a blockage of the glutamate-related toxicity in the brain. How you consider this with your data?
Well, we have to look back at what are the translational profiles in those mice, in those experiments, right? And we do have that now. What we could do is pull down pure myosin and see if proteins responsible for synaptic function are altered as well, which we can do now when we pull down pure myosin and do that experiment. The challenge with TBI is that it has to be region specific. And so we can't just pull down a protein from a homogenin. We have to do spatial proteomics, and that's not as easy to do.
Do you think that this is one of the problems we have in AD, where we use different brain areas in different stages, and we sum up, we suggest a soup of results, which is not the reality?
That's right. I like the use of the word soup. Yes, especially in late stage AD, it's really hard to determine or predict anything. I mean, there's a nice pattern of spreading, right, that has been well established. The Brock staging, for example, is emblematic of that. But there's still so much variability in copathology that's hard to narrow down, right? I mean, TDP-43 aggregates there, how are those influencing pathology, or Lewy bodies in many of these cases of Alzheimer's? There is no, I mean, identifying a pure case of Alzheimer's is very, very challenging.
And if you go a bit broader, because I know you are thinking broad through our collaboration, but also you saw it with your presentation, based on the fact that tau is more and more involved in more brain disorders with different etiology, coming from the work we are doing with depression and stress, coming to chronic pain or persistent pain in humans, coming to TBI, or epileptic changes is also considered to be mediated by tau. Would you consider this effect on translation, transcription, or one of or the other to be involved also in other diseases? So if you do believe that tau is just not an AD or tauopathies related molecule, but has to do a broader function in different brain pathologies, would you consider this, or what do you think is happening? And do you think that in all these different pathologies that tau may be involved based on the preliminary preclinical as well as clinical studies, do you think that tau impact will be different on translation transcription in different brain areas or brain diseases?
I don't know. It's possible. I actually think tau is a fundamental protein that facilitates selective translation. For example, heat shock proteins are going to be produced more when there's a result of stress, right? And I think tau helps promote translation, transcription, translation really of these heat shock proteins to promote refolding. And I think that is the case for every disease. These proteins are so fundamental to cell function that they need to work very carefully. But again, long-term expression of these proteins is going to lead to different problems and different manifestations. In frontotemporal dementia, the pathology is more frontal and temporal. You get a different manifestation as it is in the hippocampus for memory, or different brain regions for different things. Now, why those brain regions, it's difficult to say. Could it be preceded by a particular head injury for a sporadic case that affected a specific brain region, perhaps? That's where I'm thinking. Since you're asking me for my opinion, I think the way we're connecting this in a sporadic universal level, a head injury, for example, can affect a particular brain region, which then is now susceptible to a pathological process or a stress that over the course of 20, 30 years can lead to devastating consequences to neuronal function.
And last question, and I will really stop. Do you think that the different isoforms of tau and the different, even, the varieties between the isoforms in the vulnerability of different areas, as you explained very well through your talk, could be responsible for this difference in vulnerability through your mechanism of translations or transcription regulation?
I do. I didn't show the work, but we've identified that the proline-rich region is responsible or very important for its interaction with ribosomes and RNA, but also the microtubule binding region. So when exon 10 is spliced out and you get a 3-repeat tau instead of a 4-repeat tau, I expect that their interaction dynamics with RNA or ribosomes are different. And so I think a difference in these isoforms leads to a different regulation of translation.
Because that brings us back also to, and I pass the word to Ioannis Charalampopoulos, who is using IPS, and depending on the audience using IPSCs and the human platforms, is the fact that, for example, you never have, you know, a real tau pattern in IPSCs if you don't have 4R tau, because most of the human IPSCs that they are trying to have from AD patients, they have 3R, where you really need a 4R. That's what I'm asking, because sometimes also I think we mislead ourselves by using also you know, wrong models.
Well, I mean, you can use iPSCs for modeling Pick's disease, which is primarily 3R tauopathy. But even Pick's, they make both 3R and 4R, right? So those are limitations in the models. I agree with you.
Ioannis?
Any other question from the audience, perhaps, now that we have some time? No? I had just one question, Josh, perhaps I missed it. In this in vivo experiments using the GSK inhibitor for PERK, okay,
Yes.
you said that you observed an increased neuronal function, also an increase in size in several areas. So how you connect this, you know, function of GSK, specifically on PERK inhibition, with so broad changes in neuronal function or size, or, you know, all this kind of stuff? But is there a link between this?
Well, we found that the inhibitor interacted with more than 20 kinases. So it's hard to say that it's only PERK. We know that the drug had a benefit. The specific pathway is unclear. So here's what we know. We know that it's not through tau, right? Tau levels and pathological tau levels were the same, which I find very important, because, and I'll reiterate,
really true.
it tells us that a patient who's already undergoing the disease process with pathology can still experience benefits with the treatment. Oftentimes in the field, we say, well, we either detect it early or it's too late. No, there's an opportunity for folks who are already in advanced stages of disease where we may be able to mitigate the toxicity of downstream effectors. Which downstream? That's a great question you're asking. For example, translation is rescued. We have other data that we published that I didn't show today, where there is less RNA oxidation with the drug. There is an improvement in, there are a number of pathways that we identified that could be responsible for the improvement.
Right.
Did I answer your question?
Yes, yes. Did you see any relevant to, you know, to growth factors related pathways or LTP inducers or things like this? Perhaps?
Because that was fitting with the synaptic atrophy and malfunction.
Yes,
that's why
true.
Because Ioannis is working with the TRK and all these trophic pathways.
Correct.
Affected.
For example, upregulation of BDNF or molecules like this, which have a broad effect, you know, in all these functions, could be of central point.
With the proteomics, we looked, but those factors were not significantly different between our controls and in AD and in fact, in Parkinson's, they were not significantly different. It came down to those 4 proteins, which now need to be validated.
Okay, guys. Great talk, great discussion. I'm sure the video will be followed by many YouTube people as well. It will be on our website. If not, if people that would not see today, they will follow it, and I'm sure you will have a lot of emails coming from different collaborators. So thanks again for being with us. It was great.
Thank you so much for the invitation. Hopefully next time we meet, we'll be in person. Yeah.
And I know you're also organizing a very good series of seminars in University of Florida. So I think that if some of them could be, open, a lot of people from students to researchers, clinical, would like to follow some of you. Are very interesting. I've been part of that, and I really enjoyed it. So I invite everyone to join us, and if it's okay with you, I'd like to share the link with you so that everybody can be invited.
That's perfect.
It will send to me, and then we can go to the others also for the HIAAD. Okay. Thanks a lot.
Thank you, Josh.
Have a nice night. See you next month, guys.
Good night.
Thanks
Thank you
again for being
with us.
Good night.
Bye.
Goodbye.